Okay, so this is a question that I didn't explain in the video lecture, but is explained in your textbook. So I recommend you again to read your textbook. Polysums may be defined as, and they give you these following options. So read it and think about it. So if you read your textbook, you will find that the polysums may be defined as a group of ribosomes that they are working together very close to each other and they are doing translation very close to each other. So remember, it's a group of ribosomes that are working together, and the other answers are wrong. So, for example, microfilaments and microtubules. This is related to the cytoskeleton, so this is not polysomes. Lysosomes is a structure in, in eukaryotic cells that is in charge of in some way digesting some of the waste products. So this is not the answer group. And so the polysomes and lysosomes don't have any relationship. Group of chromosomes is not called lysosome. And peroxisomes, they are have a relationship with the lysosome, also um, in charge of uh, digesting some of the waste materials. But again, there is no relationship with the polysome. So the only answer, and if you see this schematic, go to your textbook uh, to study what is a polysome and to know more functions about it. So this is an easy question. I'm asking you what type of bonds hold the molecule of gRNA, the transference RNA, to have a three-dimensional shape. So here, very easy. In the same way that the DNA is held by hydrogen bonds, here the tRNA is held by hydrogen bonds. And so the answer will be hydrogen bonds. And if you remember, the other ones are wrong because so A is correct and B is incorrect because the peptide linkage are just joining amino acids to form peptides or proteins, and the hydrophobic interactions help the proteins to have a better shape, but not the tRNA. And covalent bonds and ionic bonds are not related to the tRNA. So the only option is A. Now this question, the function of tRNA during protein synthesis is one of this one, and we already studied it. So now try to answer this question. So remember the tRNA during the protein synthesis deliver the amino acid. So here you can see the amino acid to the proper site during protein synthesis. And I'm going to show you a small video that talk about that.
A tRNA is an adapter molecule that can read the genetic code and bind with a specific amino acid. A tRNA has 3 loops and a tail. The 3 loops are called anticodon loop, D loop and T loop. The anticodon loop is formed of 5 base pairs. It binds to the complementary bases on the mRNA. The D loop is a 4 base pair stem that binds to amino-acyl tRNA synthetase. The D loop derives its name from one of its bases called dihydrouridine. The T loop is a 5 base pair stem that facilitates the binding of the amino acid to the A site. The tail is the 7 base pair amino acid acceptor end of a tRNA. The amino acids bind at the CCA base sequence. The tRNA that initiates translation is called an initiator tRNA. It always carries the amino acid formaldehyde methionine or F-met. A tRNA
Okay, so now we know very well that the answer is A. So this is the answer. And we know that it doesn't guide the ribosome subunits out of the nucleus through nuclear pores. This is, yeah, that this is not the function of the tRNA. Attached to the messenger RNA to a small subunit of the ribosome. These are our proteins, the ones that are doing that. You can watch that in the video lecture, but this is wrong. Process the messenger RNA. This is not the function of the tRNA and transcribe the messenger RNA. So all these ones are wrong. And the answer is A. Okay, so something very important that takes place during transcription and translation. The first step is that when you have a sequence like this one, in order to transcription to occur, there should be a specific recognition sequence. And I want that you, if you remember, if you have a DNA and you're going to sequence here, you need a protein that attach here, and it should detect this DNA region to go here um, and start synthesizing the new sequence. So I am asking you about the name of this section of DNA. And if you remember, that section is called a promoter, and a promoter signal the initiation of RNA synthesis. And here one of the famous points is this Tata box. And the promoter is a region of DNA located near to the transcription starting site. So here you can see the starting point here, and here you can see the promoter. And as I said, in the beginning you have this sequence of Tata box. So answer will be promoter, centromere. It will be here, this region in the middle of the chromosome. Introns are wrong because the introns, if you read your textbook, are the sequence of DNA that are added and they are removed before going to transcription. So before we used to call them junk DNA because they didn't sequence anything. Now we know that they are involved in the regulation, many of them. Exons are the sequence that are used, that are the one that are going to form the proteins. And this is a codon that is not involved in the process, is not involved in this sequence, that is in charge of something else. So definitely all of these one are wrong. And here I have a video that they talk about that.
Each gene consists of exons and introns. Exons are the sections of the DNA that contain the instructions for making a protein. Introns are those sections that do not code for protein synthesis and do not carry genetic information. It is common for intron and exon borders to be delineated by the CG nucleotides next to each other in an RNA sequence. The introns will be edited or spliced out to form a final version of mRNA to be translated. The resulting exons are those sections of the DNA that eventually make proteins. Even now, we are not sure why we have introns and what their role may be.
So here I am asking you about which of the following, so I'm a catalyze, so I'm asking you about enzyme, the linkage between the ribonucleotides. So I'm asking you about ribonucleotides. It will be related to RNA to form RNA during the gene expression. So you have the following enzymes, and now you have to pick one. So if you remember, in the beginning we have a promoter, and in the promoter region, the RNA polymerase binds there, and then there is an unwind of the DNA, and then there is initiation of the RNA synthesis. So the answer will be A. Definitely, I don't think that there is an RNA ligase. There is a DNA there is a ligase. Ribosome is an interesting enzyme that is formed by RNA, but there is no relationship here between the linkage of ribonucleotides to form RNA, so this is wrong. The rubber transcriptase is a very interesting enzyme that we generally talk about, that we go from DNA to RNA. There are some viruses that do go in the opposite direction, and this is why you have this reverse transcriptase. And this tRNA is not an enzyme, so definitely all of this one is wrong. So the answer will be A. And if you have any doubt or any question, just let me know and send me an email. Have a good day and good luck in your final exam.